Welcome to Module 3. We're going to be moving into animal structures, homeostasis, form, and function. So today we're going to tackle animal form and function, specifically in the realms of tissue types, bioenergetics, positive and negative feedbacks, and body planes. So there should be a lot jam-packed into this lecture. I do recommend reading your textbook sections to give you more insights into everything we're briefly covering in this lecture. First, let's discuss symmetry. This is something you've probably already noticed about animals in the wild as well as ourselves. Many different animals display different types of symmetry, and most notably, the sponges actually display a type of symmetry called asymmetry. So here we have on the left the typical form of a sponge that's found in the ocean. There are some examples in the lab that we'll get to view during coming labs, but sponges have an asymmetric body plan, meaning you cannot find a mirror image no matter how you cut this sponge. By contrast, the sea anemone and jellyfish, they display what we call radial symmetry. So you can cut many different planes about their central axis, and it will always be a mirror image. And that's because they're basically like a cylinder. They display radial symmetry. There's a special type of symmetry that's radial in nature called pentaradial symmetry, or 5-point symmetry. This is what we see displayed in our sea stars and our sea urchins. These are our echinoderms, if you remember back to our fossil lab. They display 5-point symmetry around their center region here. It's a special type of symmetry because you can't really cut them in half and have a mirror image. Instead, you have to draw a plane out from their center. And probably the most common type of symmetry that you might be familiar with is called bilateral symmetry, and that is what you and I display, as well as many of our furry friends around, where you have a dorsal, a ventral, a posterior, and an anterior plane, and you can just slice the animal right down the center about their nose here. A standing vertebrate animal can be divided by several different planes. So here's a look at a figure inside of your textbook. So before we were using the example of the goat to demonstrate bilateral symmetry, but there are actually different planes by which you can cut humans and animals. We have the sagittal plane, which is the left-right plane. We also have the mid-sagittal plane, which divides the organism by left and right, but make sure that these left and right pieces are exactly equal to each other. So that is what we know here. We have the frontal plane, which is the front back plane. So when we put this through our midline, we have a front section and a back section. And then we have the transverse plane, which is also called the horizontal plane or the cross section. This divides us into upper and lower portions. So here in the pink, we have the transverse plane in humans, and you can have a number of different transverse planes or cross sections by which you can divide the human or the goat body. So notice the orientation is different because the goat here on the left is a quadruped. It uses 4 feet to walk, whereas humans are bipedal. We use 2 feet to walk. This would look the same if the human were to get into all fours or tabletop position, if you are a yogi like me, demonstrating the different planes about the midline. In addition to symmetric axes, we could also talk about form in the terms of shapes. We've already talked about this form before, but we've called it something else. We called it a torpedo shape. And that's what we see displayed by our sea creatures like dolphins, fish, and whales. But this particular torpedo shape has a special name called fusiform. This fusiform shape allows these animals to speed through water, reducing the drag that they have placed on them, allowing them to move through water like a bullet. So when an animal is able to generate thrust that's greater than the drag that it experiences, this is what we know as swimming. And the fusiform shape is the best shape in the water to facilitate swimming and increase that thrust. This shape allows dolphins, for example, to achieve speeds up to 25 miles an hour. On land, however, drag is not really that big of an issue. Instead, land animals have to overcome gravity. The thing is usually not drag, it is gravity. So for example, most adaptations for birds are for flying through the air. It's not drag that they're trying to overcome, it's gravity that they're trying to overcome. And that's because mainly air is less viscous than water. And so if you take a look at the skeleton of a bird, you'll notice that they have hollow bones. And that's because it allows them to have a very, very light skeleton that they can then cruise through the air with. And most land animals can actually travel faster than creatures in the sea. And we'll see that on the next slide here, where we just have a table from your textbook, table 33.1, that relates to the maximum speed of assorted land and marine animals in kilometers per hour and miles per hour. And you can see by and large that our friends on land are much faster than our friends in the sea. Moving into what the outside of these creatures are, most animals on Earth actually have exoskeletons. There are many, many more insects on this planet than other things. And so the creatures that have the feature of an exoskeleton include insects, but it also includes centipedes and spiders and scorpions and crustaceans. So there's a lot of biodiversity out there that contains exoskeletons. These exoskeletons have special ingrowths in them called apodems. And these apodems are where muscles are attached. So kind of like you and I have tendons, the creatures with exoskeletons have apodems. And what's interesting about this feature is that in order to grow, the animal must first synthesize a new exoskeleton underneath its existing exoskeleton. And then once it's ready to encompass that new exoskeleton, it must molt or shed the existing exoskeleton. And that's why, for example, right now, you probably see a lot of casings for cicadas. That is them shedding their useless old exoskeleton because they have new ones. And that material that is composing the exoskeleton is made up a substance called chitin. C-H-I-T-I-N. And we first heard about this substance called chitin in Module 1 when we were discussing the composition of cell walls within fungi. And so the exoskeletons of our insect friends and our scorpion friends, they also have the same substance that makes up the cell wall of fungi called chitin. The weight of the exoskeleton material usually limits the overall size that the animal can achieve. And we'll talk about that in the next slide as well. So whereas the exoskeleton really limits the size that an insect or centipede or spider can achieve, those organisms that contain endoskeletons are less constrained by that. They have other constraints around their size. So you and I, we have endoskeletons. So do most of our furry friends like elephants and dogs and goats and cats. They all have endoskeletons. Their growth and our growth is constrained by what is called the cube square law. And this states that as an organism grows in size, its volume, meaning the space inside, grows faster than the surface area. And this is what we call allometric scaling. I have a picture of JBS Haldane over here with his glorious mustache. Haldane is one of the founders of what we call neo-Darwinism, or what you and I have called the modern synthesis. So taking Darwin's ideas and plugging them into Mendelian genetics. But Haldane in 1928 wrote an essay called On Being the Right Size. He stated that large animals do not look like small animals. So for example, an elephant can't be mistaken for a mouse that's scaled up in size. It simply doesn't work like that because of what we call allometric scaling. And this happens both among species and within the same species. So in his 1928 essay on being the right size, he points out with a specific example of giants that we have in our children's books. And he said, consider a man that's 60 feet high. Every square inch of giant bone had to support 10 times the body weight borne by a square inch of human bone. As the average human thigh bone breaks under about 10 times the human weight, the giants would have broken their thighs every time they took a step. So the creatures, the giants, the Godzillas that you see in movies, they simply could not be that large simply because of the amount of weight that is being asked for their bones to support is not going to happen. So this wraps directly into what we call bioenergetics. And this is what this graph is showing here. As you scale up in body mass, in kilograms, for example, your total metabolic rate in watts has to increase because you're supporting more and more biomass compared to something that's pretty small. The amount of energy expended by an animal over a specified time is called metabolic rate. And the rate is usually measured in joules or calories or kilocalories. Metabolic rate is estimated as the basal metabolic rate, also called the BMR, in endothermic animals at rest. So you and I have what's called a BMR. For women, it's a little bit less than for cisgendered men. I, for example, only require, apparently, I just learned this, about 1,300 to 1,500 calories a day to maintain myself, whereas my husband would require a little bit more. The term is different when we're talking about ectothermic animals, so things that are not considered quote unquote warm-blooded, but we're not going to use those terms. I'll introduce new terms in a couple of slides. And so in our ectotherms, the standard metabolic rate, or SMR, is used. And even with insulation, it takes so much energy for endothermic animals to maintain their constant body temperature. So even though I have this wonderful thing called homeostasis and thermoregulation, it takes a lot of energy to keep my body temperature where it's supposed to be. But if you take a look at something like an alligator, that's an ectotherm, that has an SRM, or standard metabolic rate, of only 60 calories a day. Okay, so whereas for me, it takes 1,300 to 1,500 calories, it only takes the alligator about 60. So we've mentioned this thing called thermoregulation. And there are two main types of animals. We have ones that are going to regulate their body temperature, their internal body temperature, and those that do not. So a nice example here of a regulator is our bobcat. These bobcats are animals that use internal regulation mechanisms to maintain constant body temperature. So this is just like any other warm-blooded animal like you and me. It's able to maintain constant internal body temperature. They're also known as homeotherms. So regulators are also endotherms and homeotherms. Those are all synonyms for the same thing, to mean that they are able to regulate their internal body temperature. By contrast, we have other creatures such as the snake, which are typically called colloquially cold-blooded animals, but those terms are now falling out of favor. Our snake here is called an ectotherm or a conformer. And conformers are animals that allow its internal conditions to conform to external conditions. So instead of having those internal mechanisms in place to maintain a constant body temperature, it instead conforms to the ambient temperature around it. We can see this in an XY graph where we're plotting on the x-axis ambient temperature increasing to the right and body temperature of the organisms increasing along the y-axis here. As we go from cooler to hotter temperatures, this bobcat really doesn't change much. It is able to regulate its internal body temperature pretty easily. Whereas this snake, it responds very strongly to changes in external conditions in that ambient temperature here. We can also call conformers poikilotherms, and herpetologists, people that study snakes and reptiles, love to use the term poikilotherms. So you are welcome to use any of these terms to mean animals that allow internal conditions to conform to external conditions. Let's move into something called torpor. Torpor is a physiological state of decreased activity and decreased metabolic rate. This is usually in response to extreme lows in temperature or inaccessibility to food. So there's some sort of extreme pressure in the environment. Either it can't get warm enough or it can't find enough food, and therefore it goes into a state of torpor or decreased metabolic activity. When we have a period of prolonged torpor, this is what we know as hibernation. And so we have little mouse here, and bears are most notable hibernators. But I bet you've never thought about what a frog does during the winter. So we're going to watch this little video that discusses what frogs do during the winter months.
The wood frog has an astounding ability. It can produce antifreeze. This seemingly ordinary amphibian can survive at temperatures as low as 3 degrees Fahrenheit and then come back to life. Winter is coming, and the wood frog prepares for the big freeze. It starts to produce huge amounts of urine. But unlike us, it doesn't drain this waste. It stores it in its blood. As the first ice crystals creep over the frog's skin, an incredible transformation begins. The water in the frog's blood starts to freeze. The ice sucks the precious water out of the frog's cells. To prevent full-scale dehydration, the liver begins to make large amounts of glucose. As the sugar mixes with the urine in the blood, it creates homemade antifreeze. This concoction packs into the frog cells, propping them up. The antifreeze prevents too much water being drawn out of the cells by the ice outside. If they lose more than 60% of their water, the process becomes irreversible. The cells will crumple and die from frostbite. The frog's antifreeze keeps him alive. The wood frog enters a state of suspended animation. Its internal organs and metabolic activity grind to a near halt. The lungs stop working, and the heart eventually stops beating. The wood frog can survive for months with an incredible 2/3 of their body completely frozen, to the point where they are essentially frogsicles. After a grueling 8 months, spring arrives. The temperature's warm and the ice starts to melt. Hidden within his frozen tomb, the wood frog also begins to thaw. Water slowly flows back into his cells. They rehydrate and return to their original shape. Within 30 minutes, the heart restarts and the blood begins to flow. the wood frog comes back to life. After 2 days, the frog heads out to look for some food, a mate, and a much-awaited pea.
So now you know. All right, so this all gets back to the idea of homeostasis, which we've touched on at the beginning of this course, but let's delve into the details of what drives it forward. So homeostasis is used to maintain a, quote, steady state or internal balance regardless of external environment or conditions or shifts. So in humans, we display many instances of homeostasis in regards to our body temperature, our blood pH, also our glucose concentrations. And all of this is supposed to be in a healthy individual maintained at a constant level. The maintenance of these things at a constant level are due to what we know as positive and negative feedbacks. These all have to do with a set point. So a set point in glucose concentrations, in blood pH, or body temperature. And the set point is a particular value that's seen as a target, the thing that we need to shoot for and maintain in our body. A stimulus detected by a sensor is going to trigger a response if we determine that our body temperature is too high or too low, our blood pH is too high or too low, or our glucose concentrations are too high or too low. This response is aimed at returning the variable back to the set point. A really great example of this that's probably easy to bring to the front of your mind is your thermostat in your home. You set it to a certain value, probably 72. Some people have opinions that are very strong about what their thermostat should be set to. So they set it at a particular set point, and then the sensor inside of the thermostat is going to register if we are too low or too high compared to that set point. If we are too high, we're going to blast out cool air to bring it back down, or if we're too low, we're going to blast out warm air to bring it back up. And very much in the same way, our bodies do the same thing. A specific example of a negative feedback is presented here. A negative feedback dampens a stimulus. This shuts off the response and allows the system to self-stabilize. An example of this is shivering when you are cold. Here we have the stimulus that affects the sensor, that affects the control, that affects the effector, going back to the stimulus. So here, when our body temperature exceeds 37 degrees Celsius or 98.6 degrees Fahrenheit, the nerve cells in our skin and our brain are notified. This then triggers the temperature regulatory center in the brain, and then this promotes the sweat glands to activate throughout the body, which then provides a feedback back to the stimulus here. By contrast, talking about positive feedbacks, this intensifies a stimulus. This ramps up the response to achieve a defined endpoint. A really great example of a positive feedback in humans is the example of childbirth. The body wants to get back to an endpoint of not being pregnant, because if we hang on to that fetus for too long, it spells disaster for both the fetus and mother. So here we have oxytocin that stimulates uterine contractions that pushes the baby forward. That baby's head is going to hit upon the cervix, and then nerve impulses from the cervix are transmitted to the brain. The brain then gets triggered to produce what we know as oxytocin. This is a hormone that is carried in the bloodstream to the uterus and again produces another uterine contraction. So in this way, we have this positive feedback loop, ultimately to achieve the endpoint of no longer being pregnant. To drill down on positive and negative feedbacks, please access the TED Ed video done by the Amoeba Sisters.